we are starting Unit 5, which is a unit on heredity. This unit is actually also called genetics. And this unit is sometimes done in a different order. So for example, sometimes this unit is taught after DNA and protein synthesis. Sometimes this unit is taught, like we're doing it, before DNA and protein synthesis. The term genetics refers to a gene, which is a section of DNA that codes for a protein that acts to create a trait. So, genes are sections of DNA that code for proteins made of amino acids these proteins act to create some sort of trait. We didn't start using the word genetics till we knew that it was DNA that was actually giving us our traits. For a very long time, we weren't sure what was causing someone to be tall or short. What was causing someone to have blue eyes or brown eyes or green eyes or hazel eyes. These things just weren't clear to us. What we did know very early on in the history is that somehow we were inheriting, or somehow traits were being passed from parent to offspring. But the problem is that we didn't really know how they were being passed. Because especially if we looked at humans, human heredity is not very clear. So you could have a tall parent and a short parent, and the child doesn't come out in the middle. The child comes out really tall. So we run into all these problems studying human heredity. What we can do is just identify the word heredity or inherit. It means to pass from parent to offspring. So in this unit, we're going to look at some very basic examples best seen in Mendel's pea plants. We'll look at how that applies to some human traits, and then we'll look at the complexity of human and other organism traits. We do want to know that there is something that has to do with parent and offspring. Of course, we actually have a fair amount of knowledge about sexual reproduction. So we're already clear that sometimes variation is going to occur. However, we are going to be able to identify certain patterns that are repeatable. So certain repeatable patterns. So certain traits. This is where we really need to know the word trait. Certain traits follow rules. Of course, some traits we're not even sure are entirely genetic. So when you looked up nature versus nurture, you saw that some traits are purely genetic. So for example, this is looking at the inheritance of a trait that is going to be this is going to be a trait that is actually dominant. It doesn't skip any generations. And so what we see is that some traits, for example, Huntington's disease, like you saw yesterday, the very first gene in humans that was identified was actually for a brain disorder called Huntington's disease. So some traits are purely genetic. Other traits are purely your environment, your nurture. So for example, the language that you speak. That has nothing to do with your DNA and everything to do with your environment and what you learn. And then others are a combination. So we're going to kind of try and balance all of that. But a trait is some sort of characteristic. And this characteristic can usually come in certain forms. So one of the things about studying genetics first is we don't have all of the DNA and protein background that we might need. And so this was actually one of the problems in studying human heredity, is we actually, for a very long time, didn't have all of the information. So first, to study inheritance, you really needed to understand what is being passed or inherited. Now we know that that what is DNA, but for a very long time scientists did not know that. Another problem that's kind of specific to humans is actually the lifespan. Because humans have a really long lifespan, it's actually a long time between generations. So when you look at a family tree, or you look at a pedigree, the one above that showed you traits in a family tree, it's usually at least 20 years between generations. We also have a long gestational period. It's 9 months from conception to the birth. But even after birth, certain traits don't show up right at birth. So rolling your tongue, you can't ask an infant to roll their tongue. And so we do see that studying humans is very difficult. It's also a little bit harder to intentionally breed. So if we want to do some genetics experiments, we don't have a lot of humans who are eager volunteers to say, hi, I'd be happy to be part of a breeding experiment. Not really going to happen. So we are going to see that instead we're going to use what we call model organisms. And these models are going to represent studying all life and how DNA is inherited in all life. So pea plants, even though we have very little in common, the pea plants actually do provide a little bit of information about how DNA is inherited. One big one that we're actually going to see, and will do this experiment as well, is with fruit flies, Drosophila melanogaster. A lot of people say, well, I don't have anything in common with a fruit fly. Humans actually have thousands of genes in common with a fruit fly. And so we'll look at fruit flies and we'll do some investigations with them as well. So as we start this unit, we just have to be very aware that there are certain traits that we don't currently have an answer to how they are inherited. We're going to start by learning some very basic examples, but we need to go back to the history of it. We really need to understand the history of how we got our very first principles. So Mendel is considered the father of genetics, and he did experiments with pea plants. As you discovered yesterday, pea plants are a very effective organism to study in genetics because they produce a very large amount of offspring versus humans who just produce usually one at a time. It is easy to control mating. So you probably saw yesterday that Mendel actually purposefully cross-pollinated. He would cut off the sperm-producing part of the flower so that he was in control with which flowers mated with which. They can either cross-pollinate, which is what we think of for sexual reproduction, two different organisms, but they can actually also self-pollinate. Meaning that you can take the sperm from the flower and the egg from the same flower. And so that's really important because that is completely controlling which genes are being passed to the offspring. He actually not only chose a very good model organism, but he kept meticulous data. So other scientists would say, oh, well, this is more likely to happen. Well, Mendel actually counted thousands of pea plants and got lots and lots of data. A final reason that pea plants were really effective to study is that their traits come in two contrasting forms. And we're going to look at those forms in just a second. But this is really important. In humans, there's not just tall and short. There's a wide range of height. And so that wasn't really clearly understood about why there was that large range in height. In studying pea plants, their traits really only come in two forms. And so that's why these experiments were really, really carefully designed. So a few words here in this part of the notes that we really want to pay attention to that might be new to us is this idea of progeny. Progeny also means offspring. Cross means like a mating. So here the question is, we've got peas with two different traits, round peas and wrinkled peas. Will their progeny exhibit one, both, or a blend of those traits? And so what Mendel did is he was crossing two different organisms and then looking at the offspring, growing the offspring into plants and looking at what the physical appearance of the plant was. Remember, that's all that Mendel could see is the physical appearance. So we want to look a little bit at this and see these titles, P generation, this means parent. So here the parents were round and wrinkled. These are new words as well, homozygous. Now the prefix homo means the same. We are actually able to use these words because we know about the actual chromosomes, which we'll get to in a second. But the words that Mendel used were pure breeding. Meaning this wrinkle plant, he crossed them many, many times so that all he ever got were wrinkled. So that he knew that these genes were pure for wrinkle and these genes were pure for round. What he saw in the F1, F stands for filial. This just means the first rounded offspring. So round times wrinkle, he got all round. So it seemed as if the wrinkle was disappearing. Then he let these particular round plants mate with themselves. And what he would see in the F2 generation, and this is where the data usually starts to reveal itself. In the F2 generation, he saw 5,474 round seeds, 1,850 wrinkled seeds. And so the ratio, this is a really important word as well, right? We can all do fractions. We know that a fraction is part compared to whole. A ratio is part compared to part. So round is 3 out of 4, but that's 3 round for every 1 wrinkle. And this is an estimation. You'll notice this is not exactly a perfect number. This is actually some of Mendel's data. And so what Mendel was able to look at is that the traits did not blend. So Mendel's data very clearly revealed certain patterns, that the traits did not blend, but they also did not completely disappear. And so what he really wanted to pay attention to is, how is it that they're re-showing up another generation later? How is it that they're skipping this generation? So one of the things that we're really paying attention to is that his data was very carefully collected and analyzed. And so what we're going to do in the kind of next part when we come back is we're actually going to analyze his data. But again, we want to see these traits that he picked out, that they come in 2 contrasting forms. And this is actually when he decided which ones were dominant and which ones were recessive. Let's go ahead and add in the word hidden. And so I'm going to stop the video here so that I can go on from here. But take a second and really look at his data. It's very impressive, the amount of data he collected and the way that he was really able to look at parent and then F2 generations.